Hello students, I'm again back here. I'm Dr. Nidhi Sharma and again with a new lecture. I hope you find my lecture very informative. Please, if you find my lecture informative and useful, please like and please subscribe to my channel. So in the previous lecture, we studied about the prokaryotic cell. Okay, and in the prokaryotic cell, we studied about the bacterial cell. This is the diagram. Are you seeing this diagram? This is a bacterial cell. Here, the outer sheath is of mucilage sheath. Then we have the cell wall. Then the cell membrane is there. Okay, we studied the function of cell wall and cell membrane. Then there is also the nucleoid. This is the nucleic acid without any nuclear membrane. Okay, so this was about the genetic material. The genetic material in the case of the bacteria, this is the bacterial cell. In the case of the bacteria, you can also find in the form of a plasmid, which is a round genetic material. Okay, so this was all about the bacterial cell. We will continue further. As we know, the genetic material, the genetic material, the genetic material of the bacterial cell, of the bacterial cell is called as, the genetic material of the bacterial cell is called as the nucleoid. This nucleoid contains the nucleic acid without any genetic material, without any nuclear membrane, without any nuclear membrane. Okay, so this is a nucleoid, this is the nucleic acid without any nuclear membrane. Okay, now this nucleic acid, generally it is in the form of DNA. Nucleic acid, it is in the form of DNA. We all know the full form of DNA, that is deoxyribonucleic acid. Okay, now in this bacterial cell, there are certain membranous structures also there. In the bacterial cell, there are certain membranous structures. There are certain membranous structures. Membranous structures. Okay, and this bacterial cell, since it is a prokaryotic cell, it lacks doubly membrane bounded organelles. Okay, like we have mitochondria, endoplasmic reticulum, okay, and Golgi bodies. All these doubly membrane bounded structures are absent in bacterial cell since it is a prokaryotic cell. Okay, now certain membranous structures are also found in them. Certain membranous structures, certain membranous structures, certain membranous structures. Okay, they are also found, such as the mesosomes, mesosomes. Okay, so mesosomes, certain membranous structures such as mesosomes and chromatophores and chromatophores. Okay, so all these structures are also present in the prokaryotic cell. Now first we'll study about the mesosomes. Okay, now mesosomes, they are the extensions of the plasma membrane. First point, we will say they are the extensions of plasma membrane, into the cell. So mesosomes are the extensions of the plasma membrane into the cell in the form of vesicles, in the form of in the form of vesicles, in the form of vesicles, or it is in the form of tubules and lamellae. Tubules and lamellae. Okay, so mesosomes are the extension of the plasma membrane into the cell in the form of vesicles, tubules and lamellae. Okay, mesosomes are equal to mitochondria in eukaryotes. Mesosomes are equal to mitochondria. We all know in a cell the mitochondria is called as the powerhouse of the cell. Why it is called as powerhouse of the cell? Because it produces energy. Because it produces what? Energy in the form of ATP. What is the full form of ATP? That is the adenosine triphosphate. Okay, so the mesosomes in the prokaryotic cell are equal to the mitochondria in the eukaryotic cell. Okay. Now, what are the functions of the mesosomes? The functions of mesosomes. Okay. First function is that it helps in respiration, and the cellular secretion. It helps in respiration and cellular secretion. Okay. Then second, it helps in increasing the enzymatic content and surface area of the plasma membrane. It increases the enzymatic content and the surface area of the plasma membrane Okay. Now the next function of the mesosome. Is help in the formation of cell wall. It helps in the formation of cell wall. Then the next function, it helps in replication of DNA. It helps in the replication of DNA. What do you mean by replication of DNA? When a new DNA is formed from a original DNA. Okay, new DNA is formed from original DNA, then this process is called as replication. So this was all about the function of the mesosomes. Now we'll study about chromatophores. What are these chromatophores? What are these chromatophores? Firstly, these are another membranous structure which are present in prokaryotes. These are again what? Membranous structure. Membranous structure. These are the membranous. Chromatophores are the membranous structures which are present in prokaryotes. Present in prokaryotes. Okay. So chromatophores are membranous structures which are present in prokaryotes. They are internal membrane system of photosynthetic form. Okay. They possess photosynthetic pigments. They possess, what do they possess? Photosynthetic pigment. Photosynthetic pigments. Okay, photosynthetic pigments. These pigments are light reflecting. These pigments are light reflecting. Okay, these pigments, they are what? Light reflecting. Now the next topic which we'll study about is the flagella. Can you tell me what is this flagella? What is this flagella? Flagella, it is the locomotory organelle. Locomotory organelle which is present in bacteria, which is present in a bacterial cell. So this flagella helps in the locomotion of the bacteria. Okay, now this flagella, it is in the form of, it is a thread-like appendages which extend from the cell wall. It is what? It is a thread-like appendages. Thread-like appendages. Okay, which help in the locomotion, and it exists and it extends from the cell wall and extend from cell wall and extend from the cell wall. Okay, now the single flagellum, the single structure is called as, single structure of this locomotory organ is called as flagellum. It is called as flagellum. The flagellum. Okay, now each flagellum is about 1 to 7 micrometer long. Each of this flagellum is 1 to 7 micrometer long. Each flagellum is how much? 1 to 7 micrometer long. 1 to 7 micrometer long. Okay, it is long and it is covered by a protein coat. And covered by protein coat. Okay, so each flagellum is 1 to 7 micrometer and is covered by the protein coat. Now we will differentiate bacteria according to the arrangement of flagella. If this is the bacterial cell and the flagella is present around the whole cell membrane, okay, or around the capsule, then it is called as peritrichous. If the flagella is only at one end, like here 2-3 flagella are here only at one end, then it is called as lophotrichous. If the flagella is at the opposite two ends, then it is called as amphitrichous. Okay, peritrichous, lophotrichous. And if there is bacterial cell with no flagella, then it is called as ertrichous. Okay, so these are the arrangement of the flagella in the bacterial cell. If it is present whole around the capsule, then it is called peritrichous. If at one end, lophotrichous. If at both the ends, amphitrichous. If there is no flagella, then it is called as ertrichous. Okay, so this was about the arrangement of flagella in the different arrangement of flagella in a bacterial cell. Now, the bacterial flagellum, the bacterial flagellum, bacterial flagellum has following three parts, has three parts. The first one is the filament. Then the second one is the hook. Okay, and the third one is the basal body. Okay, so these are different structure. Now, are you seeing this diagram? This is the bacterial flagellum, which shows the different part. This is the filament. This portion is the filament. The filament is the longest portion which extends from the cell surface to the outside. It is made up of a protein called the flagellin. It is made up of a protein which is called as the flagellin. Okay. Then we have the hook. This hook is curved and tubular structure. It is curved and tubular structure and made up of protein subunits. Then we have the basal body, the most complex part of the flagellum. It is the most complex part of the flagellum. So this is the bacterial flagellum, which shows its different parts. So you know, a bacteria is made up of filament, hook and a basal body. Okay. Now we all know what is the function of this flagella. This we have discussed also. Function of flagella. What is the function? It helps in locomotion. Helps in locomotion. What is locomotion? The movement of bacterial cell from one place to another is called as locomotion. Okay. So this was about the flagella. Now the next surface structures. There are more next surface structures. First one is pili, and second one is the fimbriae. Okay. So first one is pili, next is the fimbriae. Now these are also surface structures, but they do not play any role in the locomotion of the bacteria. The pili are elongated tubular structures. They are what? Elongated tubular structures. Okay. So pili, they are what? Elongated tubular structure. And they are made up of which protein? Pillin. They are made up of a small special protein called the pillin. Made up of, these are the protein. Okay, protein subunits, you can say. Protein subunits. Okay, so the pili is made up of pillin. These are small protein subunits. Okay, and fimbriae are bristle-like structures, bristle-like fibers coming out of the cell. While the fimbriae, this fimbriae is what? A bristle-like fiber which come out from the cell. Now we'll study about the function. The function. Okay. Function of firstly pili. What is the function of pili? Generally, the pili helps in forming conjugation tube. Pili helps in forming the conjugation tube. It helps in forming the conjugation tube. Okay. During the sexual reproduction of bacteria. Sexual reproduction of bacteria. Okay. So pili helps in the formation of conjugation tube during the sexual reproduction of the bacteria. Okay. And this pili helps in the transfer of genetic material between the donor and the recipient cell. Okay. Now, what is the function of the fimbriae? The function of the fimbriae, function of fimbriae, function of fimbriae, function of the fimbriae. Now, what is this function of the fimbriae? It helps in the attachment of bacteria to solid surface, helps in the attachment, helps in the attachment of the bacterial surface of the bacterial cell to the solid surface, to the solid surface. Okay, so function of this fimbriae, it helps in the attachment of the bacterial cell to the solid surface. Now we'll study the difference between pili and fimbriae. What is the difference between pili and fimbriae? Pili, they occur mostly in the gram-negative bacteria, and fimbriae occur in the they occur in both gram-positive and gram-negative bacteria. So this is the basic difference. And pili is longer and broader. Fimbriae are shorter and narrower. Pili, they are responsible for conjugation in bacteria. Fimbriae, they are specialized for the attachment of bacteria to its host. Pili, they are tubular structures, while fimbriae are bristle-like solid structures. So this is the basic difference between the pili and the fimbriae. Okay, have you understood now? Now you must be thinking, what are the gram-positive and gram-negative bacteria? This I will tell you. Gram-positive and gram-negative bacteria. Gram-negative bacteria. See, there was a scientist called Christian Gram. Scientist called as Christian Gram. Christian Gram. Christian Gram in 1884. In 1884, he find out the various type of reactions. He found out the various type of reaction, various type of reactions shown by the cell wall, of different bacteria, Okay, now what he found? He found that there were some bacteria which took up the gram stain and retained the purple color. They were called as the gram-positive bacteria. So the difference between gram-positive and gram-negative bacteria is done on the basis of the differences in their cell wall and response to the staining procedure developed by gram. So the gram-positive bacteria, what this bacteria did, that they took up the gram stain. These bacteria took up the gram stain. Okay, took up the gram stain and retain the blue or purple color, and retain the blue or purple color. Retain the blue or the purple color. Okay, and retain the blue or the purple color. Example of gram-positive bacteria. Example of the gram-positive bacteria. Example is what? Here we have Bacillus subtilis. Bacillus subtilis. Bacillus means here rod-shaped bacteria. Okay. Now the second one is the gram-negative bacteria. The second one is the gram-negative bacteria. Now this bacteria, it does not take up the stain, gram stain. It do not take up the gram stain. So it is called as gram negative bacteria, and it lose the blue or the purple color. It lose the purple or the blue color. Okay, it lose the purple or the blue color. The best example of the gram negative bacteria is E. coli. What is the full form of E. coli? Escherichia coli. The full form of E. coli is Escherichia coli. So this is the basic difference between the gram positive and gram negative bacteria. Gram positive bacteria takes up the gram stain and retain the blue or the purple color. Example is the Bacillus subtilis. While the gram negative bacteria does not take up the gram stain and it loses the blue or the purple color. Example is the E. coli or the Escherichia coli. Okay, so this was the basic difference between gram positive and gram negative bacteria. Now the next topic which we'll study about is ribosomes. This is what the ribosomes. What are these ribosomes? Can anybody tell me here? Okay, ribosomes, they are actually they are found in prokaryotes. Ribosomes are found in prokaryotes. They are found in prokaryotes. Ribosomes are found in prokaryotes and they serve a common function. What is the function of ribosome? Protein synthesis. Okay, the function of ribosome is protein synthesis. So ribosomes are small but are complex in their structure. They may be about 15 to 20 nanometer in size. They are 15 to 20 nanometer in size. 15 to 20 nanometer in size. So in the case of the prokaryotes, the ribosomes are found in association with the plasma membrane of the cell. Okay, the ribosomes in the case of the prokaryotes, they are found in association with the plasma membrane of the cell. Now the prokaryotic ribosomes are of 70S type. The prokaryotic ribosomes are of 70S type. 70S type means it has following two units, following two subunits. First one is the smaller unit, which is 30S, and another is large subunit, which is 50S, So ribosomes are found in the prokaryotes. Their function is protein synthesis. They are 15 to 20 nanometer in size. Prokaryotic ribosomes are of 70S type. They are made up of following two subunits. That is, smaller subunit is 30S, large subunit is 50S. When ribosomes are arranged in helical group, then they are called as polysome or polyribosome. Polysome or polyribosome. These terms are used when the ribosomes are attached to each other. Okay, so in each polysome there are in each polysome, okay, there are 4 to 8 ribosomes are there. 4 to 8 ribosomes are there. Ribosomes are in each polysome. 4 to 8 ribosomes are attached to a single strand of mRNA. Are attached to a single strand of mRNA. Strand of mRNA. Okay, so in each of the polysome, 4 to 8 ribosomes are attached to a single strand messenger RNA. Now this ribosome of the polysome helps in translation. Okay, help in translation. What is this translation process? Translation is the process of formation of protein, the protein from the messenger RNA. So the ribosome of the polysome help in the translation process. Okay, and what is this translation process? It is the process of protein formation from messenger RNA. Okay, so this is about the ribosome. Now there are certain inclusion bodies are also found there in the prokaryotes. Inclusion bodies are also found in the prokaryotes. They are what? Non-living structures which are present in the cytoplasm. They are non-living